applications, especially our recent ambitious project on greenhouse gas emission, which is an important topic for the whole world, I think. Yeah, and also because of oil sands industry in Alberta, so that's a pretty essential topic here in our region. And I will briefly introduce the extension to machine learning approach. And one purpose is for the validation of our prior assumptions in our stoichiometric models, and the other purpose is to integrate machine learning with mechanistic models. I will mention briefly the logics behind that. So let me first give a bit background about mathematical biology, or you can call it mathematical biosciences. And of course, we have tons of challenges nowadays. And here I mention a few of them. First one is mechanistic modeling by fundamental physical and chemical laws. So stoichiometric theory is a good approach or tool to handle this mechanistic modeling. And true model validation is nowadays it's not easy because we can see all kind of validations or comparison of model with data, but a lot of approaches have overfitting. And I will show one example that we have fully controlled parameters from experiments such that overfitting is avoided. And Xiunan has done a lot of work on the inverse method for infectious diseases. That also shows potential overfitting in a lot of infectious disease model with data. And I will also show the transient dynamics versus asymptotic dynamics. As we know, as mathematicians, almost all our tools are for asymptotic analysis, like stability of steady state, right, bifurcations. So all those tools are for asymptotic analysis. And for management purpose, if we talk to policymakers, stakeholders, in many cases, transient dynamics can be more important. So I will also show that idea in using the same model later. And we hope we can develop novel mathematical or computational tools motivated by bioscience, it's like physics did for math society. And nowadays we cannot avoid data science and machine learning. So we are in this era, and we have to confront data. And I think in many cases we have to use machine learning. The logic is that yes, mechanistic modeling is good, but for many systems, it's kind of a gray box, which means we know some rules or some mechanisms, but we don't know a lot of other rules, right? We don't know everything of a system. That's the common situation. So we hope the machine learning or data science can be integrated into the mechanistic model. So for example, for infectious disease, SIR, that's fine, those basic mechanisms inside. However, for any new disease or in real situation, there are many other factors kicking in, right? So there are many covariates we ignore when we assume something for the model to start, right? So there's a lot of missing information there, right? We don't know. So we hope machine learning can help to make that up. And yeah, I think Xiunan also did several papers on that hybrid modeling. Try to use some simple machine learning approaches together with mechanistic modeling. So I think that's the future, but we are still in a naive stage as a start. And biology is very hard, right? Very hard, really complicated. So here just the examples I always use, borrowed from Jim Elser's presentation. Kind of left side we have oversimplified food web for Northwest Atlantic. So you can see how many equations you need if you model this kind of system. And it's, of course, already oversimplified from real system. Right-hand side is within a cell. What's the metabolic pathways? Right? So it's also very complicated, even within one cell, right? If we want to model all metabolic pathways. I think now it's possible, you can do it, but just you have hundreds, thousands of equations, right? So it's kind of, if we want to understand the mechanism for some phenomena, it's kind of, we need to find the needle from this hay, right? So that's the feeling, right? Because there are so many covariates. And mechanistic modeling, if we use equations, we need to have prior knowledge. We need to have a lot of assumptions that we maybe, in many cases, we are not 100% sure, right? So as I said, okay, in modeling course, it's very easy to criticize a model because all models are wrong, but some can be useful, right? If in specific situation, and we know when the model is suitable, right, for specific questions. So the idea here is, can we make needle large enough so we can see the mechanism? It's kind of hidden mechanism, right? So normally it's hidden. So causality is always hard, right? Always hard. So especially if you see field data, we have some issue even to tell the whole story behind that, right? From data. Yeah. Then we may have make wrong assumptions, right? We have wrong mechanisms or hypotheses, right? That's all possible. So can we make the needle large enough? Then we can see the mechanism which is hidden, okay, which is hidden from phenomena or from data. Okay. So stoichiometry is one good approach. Okay, it's mechanistic and also can make this hidden mechanism visible. Okay, visible or tractable. So if we look at compare different species, okay, here just mention is the Daphnia, which is the most common species used in the lab to show stoichiometric effect, which means the key elements effect, right? So if we compare with other species, seems like this definitely has higher requirement on phosphorus, right? So you can see 80 carbon need 1 phosphorus. So compared to others, CP ratio is lower here, which means phosphorus requirement is higher for Daphnia. So we can use this species in experiments to show the impact of, we can say, limitation of phosphorus in on the different situations, right? So here I put two equations. Okay, I don't write down mathematical equations. You can, okay, you can, but to save time, I don't do that. But I think logically it's the same. Okay, it's the same. These two equations actually we're familiar, but they look unfamiliar because I use elemental computation, right? You can put more key elements, okay, not just CMP, can be more elements, okay, involved. So here we can see the first one is photosynthesis, right? So it's kind of autotroph plants use inorganic matter, right? Inorganic matter, and the light as energy source. Then they can produce more plants, and they may exclude like some extra inorganic matter if they don't need, right? So they may exclude something. And the bottom one is predator prey. We're also familiar, like it's kind of predator consume prey, then prey is gone, then they will produce more predator, right? Either through biomass growth, like, or can be through like birth, right? Next generation, plus waste. Okay, they will exclude extra elements to keep the balance. Okay, so that's about the homeostasis of elements. I'll mention that soon later in this talk. Okay. So we can write our familiar equations in elemental computation. Of course, these kind of equations will be more, I think it's harder, right? Harder to write down. It's easy to make mistakes, right? So we need to keep track of elemental cycling and the flows, right? Within these kind of systems. Right? So later I will show you one example. I don't show these two, but I think I will show one example for stoichiometric model. And normally most stoichiometric models spend some time, okay, to explain details. So I can only show maybe one or two of them in this talk as an illustration, and others just by graph or by conceptual equations like this. Okay, so why elements are important? Okay, so here, first we can compare the mismatch. Okay, so if we look at human body, you can also look at animals, right, and compare with the environment. So it can be earth crust or can be like those aquatic systems, right? So in water body. So you can compare with living organisms. Maybe one choice here we choose human body, and also one environment like elemental composition. So here we choose earth crust, the composition. So we can clearly see the mismatch because in human body, percentages of carbon, phosphorus, and nitrogen are much higher than the percentages of carbon, phosphorus, nitrogen in the nature, right, in Earth's crust. So that gives some hint. Okay, living organisms make a very discriminating selection of elements from the environment, right? Which is not good for living organisms, right? Can we have a better strategy to use elements to form organisms? Yes. So that's possible, right? That's possible. But there are also reasons why we make this composition in cells, right? So you can discuss a lot about this in the periodic table of the elements, right? So we are formed by carbon structure, right? So carbon is here, and you can imagine this silicon has a similar structure, but they can form a similar number of molecules. But we know that silicon structured molecules are less stable compared to carbon structured, right? So that's main reason, right? We want more stable case. And of course, not transition from human to robots, kind of transition from carbon to silicon. There are a lot of silicons, okay, in Earth's crust. Okay. Yeah, so there are a lot of interesting stories. We can discuss key elements. We can compare different organisms with different environment, environmental element composition, right? So the mismatch can cause what? Cause limitation, right? Can cause limitation. So when we discuss growth of human, growth of animals, growth of plants, right? They may be limited by some of these elements under different situations. We'll see. Yeah. So here is a kind of a simple energy pyramid, right? Energy pyramid. So I will use this one example here, phytoplankton, which is algae, right? So it's plants in the water, and the daphne are consumers, which eat this algae, right? And algae, of course, they grow through photosynthesis, right? Use light, and yeah, inorganic matter, so they can grow then. Heterotrophs like daphne, herbivores, or fish, or human. Of course, we always need to grab energy, right? We cannot create energy. We don't have photosynthesis. Right? So that's kind of simple idea, but that's kind of carbon flow. Okay, when we talk about energy, it's kind of carbon flow. And stoichiometry, we need other elements, which can also be essential, depending on situations. Okay, and on environment conditions. Yeah. So here we can see one example of experiments. Right, this done like 20 some years ago, which shows that in low light, there are a lot of in low light should be not much plants, right? Not much plants. And but the phosphorus carbon ratio is high, which means it's kind of less food, but nutrient rich, right, in the food. And in actual highlight, we can see the kind of opposite, right? So in actual highlight, we know that we'll get a lot of plants. It's measured by algal carbon, right? But the herbivores who consume these plants cannot survive even. They may go extinct. The reason is this curve with the circles here. So it has a low nutrient content, like PC ratio, right, in the food, right, in the food. Which means a lot of food, but trash food, right, so can be negative. So obviously, classical consumer resource or predator-prey model cannot capture this, right? Those models just say, okay, a lot of food means that's good, right, for the growth of the predator or the consumer. And we had a model, okay, so there's several models, okay, existing in the literature using stoichiometric theory and which can show this phenomenon. So if you are familiar with predator-prey dynamics, okay, the first two pieces are the same, okay. First two bifurcations, transcritical bifurcation and Hopf bifurcation are the same as standard classical predator-prey model, right? So if the light is low, then we don't have enough food for the predator or consumer here, right? Then extinction. Then the E2 is globally asymptotically stable if the light is a bit higher, right? Enough food for the consumer to grow, right? Then we have Hopf bifurcation, right? That's supercritical Hopf bifurcation. Then we generate limit cycle. So that's a classical one, right? It's classical predator-prey model can give that. But after this Hopf bifurcation, then we have something new, okay? So we will get bistability region, and we have the new equilibrium E4 can be globally asymptotically stable, right? Globally attracting when light is stronger. And when light is too strong, we have another new transcritical bifurcation, which can cause the consumer to go extinct, right? So that's E1 attracting again. But the reason is different. The causality is different, okay? E1 on the left, when K is low, it's kind of because of the food limitation, right? You can say food quantity limitation. And on the right-hand side, if K is strong, K is large, which means light is strong, then we have the extinction again because of the low food quality, right? So reasons are different, but the result is the same. Okay, consumer cannot survive. Yeah, so underlying reason is pretty simple. If we just ask what's in a cell, right? Because all organisms composed by cells. So if we look at the cell, we see that carbon supplies energy, right? And nitrogen is essential to build proteins. Phosphorus is essential to build nucleic acid, DNA, RNA. So higher phosphorus probably means higher growth rate, right? If you have higher nucleic acids. Right? But of course, you may be also like the other side, you will be more like limited by phosphorus limitation, right? So it's kind of trade-offs. Okay, trade-offs. So, and then already mentioned the classical play model cannot explain this simple experiment in the previous slide, but stoichiometric model can, right? As I have shown in the previous slide, right? And I performed the experiments like 20 years ago to treat two species of Daphnia under different environment conditions. So we can see from the left side, there are two groups of bottles, right? One is with the transparent ones without black cover, one with black cover. So that causes the low light and high light intensity on the right. Right? And it's interesting, we have two species, the Limhosi, the red one, it's an invasive species from Africa, and the Purex is the native species in North America. And we can see clear opposite competitive exclusions, right? So in low light, the invasive species excludes the native species, right? And in high light, the invasive species wins. And in low light, the native species excludes the invasive species. But of course, it takes a long time. Okay, it takes a couple of months to exclude them. Right? So the invasive species is more aggressive. Okay, it's more aggressive. So it's interesting to show this from modern perspective. So stoichiometric model can make that. Okay. So from stoichiometric model, I don't show the equations. If you have interest, you can read the original papers of these kind of stoichiometric models. So we can see that low light intensity is from data fitting. It's here on the left and the high light on the right. So the bifurcation diagram can easily show the opposite competitive exclusions. And of course, the model also exhibits the chaotic coexistence in the intermediate light intensity. Okay, so which shows on the right here as the pathway to the chaotic attractor. Okay, chaotic attractor. So that these two species can coexist. Yeah, of course, nowadays there are a lot of discussions about how to show the chaos experimentally, right? There are some ways to check that, although not 100% sure about that, because chaotic orbit, it takes forever to have different shapes, right? So we need some tools to validate that. Okay. And to do a stoichiometric modeling, we need a group of fundamental laws. Here I list some of them that are commonly used in this kind of modeling. Of course, you can find more, right? Find more from stoichiometric books. So conservation law of matter, we are familiar from high school chemistry book, right? So in normal chemical reactions, before and after, if we see check the each element, right? Each element then should be conserved, right? Conserved for each element, right? Before and after normal chemical reactions, right? So that's very useful law to check whether a stoichiometric model is correct or not. Okay, we cannot simply add a term, subtract a term. Okay. And Libby's law minimum, logically, it's very easy to understand. You can kind of this barrel here, the water level is determined by the lowest side, right? So similar idea, the growth rate of an organism or a cell is determined by the most limiting factor, right? Most limiting factor. So in case we have many limiting factors, right? And Gibbs form is a more mechanistic version for cell growth. Okay, for cell growth. So the cell growth depending on the actual nutrient contents in a cell. Okay, so it's inside the cell queue. Okay, so normally we use monod form. I think most people use monod form for resource-based growth, right? So that's a simplified version. I will mention that in the next slide. Okay, to compare with Gibbs form. So the logic is simple because the Gibbs form just says we need to grab the resource into our body first, then we can grow. Okay, not the resource outside. Okay, so sometimes they are consistent, sometimes they're opposite. Okay, I will show you in the next slide briefly. And Lambert-Beer's law, that's a well-known law in physics, right? So we can use this to quantify light intensity along water column. Okay, and in many cases we need to modify this law. Okay, in biology. And homeostasis, I mentioned that a bit ahead. Okay, so it's about the ratio of those elements in organisms is the same, balanced, or unbalanced, which means they change, vary a lot. Okay, so normally people assume strict homeostasis for autotrophs. Okay, in stoichiometric model. Sorry, opposite. Strict homeostasis in heterotrophs. Okay, heterotrophs, which means in heterotrophs like animals, bacteria, we assume the ratio of the elements is almost fixed. Okay, almost fixed. But in plants, that varies a lot. So the plants will not follow this homeostasis assumption. Okay. So in these two papers, we just want to check when this assumption makes sense, when it does not. Okay. So you can take a look to see and when we can use this kind of assumption. Okay, to simplify the model. So this strict homeostasis assumption can dramatically simplify stoichiometric models. And here we a few years ago, okay, with my PhD students, we wrote a paper to compare the most popular way to model resource-based growth. That's called monod, right? Monod form. And we compare that with Gibbs form. We think that's more mechanistic. And many people may say, oh, they are the same. Okay, because monod may be a good approximation for Gibbs. Yes, in some cases, yes. Some cases not. Okay. So monod form is from this graph easy to see. Okay, so Gibbs has like consumption first, then growth. Okay, monod just have one step. If you have the resource in the environment, you can grow, right? So that's the monod assumption. So before any student working on stoichiometric modeling, I always ask them this simple question. Okay, on the ideal situation, I ask them, okay, is more nutrient available in media, which means in environment, is good or bad for algae? Okay, so ask this simple question. And some students may answer first, some students may answer second. So if they don't know stoichiometric modeling or Gibbs form, then they will answer the first one, I think. So more nutrient in media gives more, in media means in environment gives more available nutrient for phytoplankton to grow. Okay, so that's good for algae. That's the monod form. Okay, so if you have in the environment more resource, that's good, right? That's good. And if you have the conservation law and the Gibbs form in your mind, then you may answer true. Okay, due to the conservation law matter, more resource nutrient in media in the environment means less nutrient for algae to grow, right? Because they will have less nutrient inside if under these ideal assumptions. Okay, so it can be opposite, right? Can be opposite. So yeah, I think it's interesting to discuss this kind of logical question. Okay, so in the paper we show people through this paradox. Okay, so it's resolved, and we can show people when we can use monod form as a good approximation. Because if both give similar results, we should use simpler one, which is monod, right? But if monod gives opposite result, then we have no choice. We have to use Gibbs form, even though it's far more complicated. Okay. But otherwise, we don't get the correct result or cannot show it, the phenomenon we want to explain. Okay. Yeah, so here I will show you one example using equations. Okay, so I think I need to show you some equations as example. So try to simplify this story in the next slide for the model. I don't show you the whole model, but the sub-model, okay, to show a stoichiometric idea. So also like 28 years ago, I had this paper modeled for bacteria and algae using stoichiometric theory. Okay, in the empirically means the top layer of the lake, right? So it's a warmer layer, and it's quickly will mixed overnight. So we can use integration to simplify the spatial structure. Okay, so it will be integral OD, and the bottom layer, of course, more complicated because quiescent. So we have to use a PD, right, to model it, and that has been done. Okay, in a later paper. Okay, in a later paper. So the full model was published, okay, several years ago. Okay, for the connecting this empirically using integral OD and the hyperlinear using the PD, right? And we need to couple them nicely, right? Okay, let's look at the model. So this is a sub-model. Okay, so I just show you the algae piece. Okay, algal piece of the model without bacteria to illustrate the idea, and I will use this to show the idea of transient dynamics as well. So the model is simplified under the assumption, okay, quickly well mixed, so we can avoid the PD. Okay, so three variables. A is Lg, here measured by carbon, and Q is cell quarter, is kind of phosphorus carbon ratio, and P is dissolved inorganic phosphorus in the media that Lg can use. Okay. So here, of course, the situation will be more complicated because we have the cell quarter explicitly, the phosphorus carbon ratio, right? So the uptake of phosphorus from environment will be added to the cell quarter, not to Lg, right? Lg. And algal growth will depend on Q, okay, Q cell quarter. That's the Gibbs form, right? So this is the Gibbs form, okay, Gibbs form, 1 minus the minimum cell quarter over actual cell quarter of Lg, right? So you can see that when algal cell quarter Q over Qm, this is positive, okay, it can grow, right? And this one is follows the Lambert-Beer's law and also the well mixing assumption. Okay, so I follows the Lambert-Beer's law. I in the light intensity at water surface, right, decays over the depths, right, and KBG is background light attenuation coefficient. And here the Lambert-Beer's law is modified because we have organisms Lg inside, right? So it's modified by A. So we have Ka here, K times A here. So K is kind of specific light attenuation coefficient. A is algal density, right, measured by carbon. So this quantify this light intensity along depths. Right? And we have this kind of monod form, right, to quantify the light uptake efficiency and take average over the top layer, the depth of the top layer, right, M-median. So Zm is the depth of the M-median. To simplify the PDE part, okay, to be integral form, okay. And multiplication is a modified version of Liebig's law minimum. So we can use minimum of 1 minus Qm over Q and this 1 over the time integral, right? So that's similar actually. So we can argue that this kind of mathematical simplification, okay, of the minimum function. So we can use minimum function or we can use multiplication, okay, to mimic the Liebig's law minimum. And the respiration loss, so CO2 is lost, right? Is lost when Lg has a respiration, right? So because Lg here, it's measured by carbon, right? Measured by carbon. So respiration, you will reduce the carbon biomass in Lg. So if you look at algal cells, they will shrink, right? They will shrink over time. And algal sinking and water exchange. So nu is the sinking rate, and D is water exchange. Both have units, kind of meter per hour or per day. So it's kind of, in general, just the length per time, okay? Length per time. So for the unit balance, when you divide by Zm, which is the depth, okay? And also this was tested in experiment, okay? So because the event for sinking and water exchange occurs at the boundary, okay? Either at the surface or at the thermocline. So the depth is, if it's larger or it's deeper, the M-median, then it's harder to have this sinking or water exchange. Similar idea for this phosphorus input or water exchange rates. And the phosphorus consumption, so phosphorus consumption by Lg, so it's kind of rho, it's per unit times A, right? And per unit one will get into the replenishment rate of cell quarter. So this rho here is can be written as this form, right? Uptake term P over N plus P times this control term, okay? So when the cell quarter Q reaches the maximum cell quarter Q capital M, it's 0, okay? They cannot uptake any nutrient anymore, right? So if Q reaches the minimum Q little m, then it's 1, okay? So it's the strongest uptake, right? Strongest uptake. And the last one looks similar, a similar term like the growth term, right? So it's the cell quarter depletion term, which is proportional to the algal growth term. They look similar if you compare the growth term and this depletion term of cell quarter. The only difference is A here becomes Q, right? Becomes Q. So that's just simple unit conversion, or you can double check A times Q, take derivative with respect to T, product rule, these two should be cancelled, right? Because one cell divided into two, the total phosphorus in those cells is the same, right? The same. So each cell will just have half of the phosphorus, right, from original cell, right? So you can double check A times Q, take derivative, product rule, these two should be cancelled, okay? So it's kind of mass balance, right? So conservation of matter, right? Yeah, so we can use conservation of matter, right, to check model is correct or not, okay, for stoichiometric modeling. Okay, so this is one example. I think it's simple enough to show, but still much more complicated than a classical population model, right? And it's very easy to make mistakes for this kind of modeling. Okay, so to save time, I just show you main math results for this subsystem. Okay, I cannot show you the result for the whole system, but I show you the results for the subsystem ergodynamics, right? So we can obtain interesting dynamics, right? Mainly symptotic dynamics, right? So like stability, global symptotic stability, right? So here we can use interesting theory in the monotone dynamic system, right? So the model looks complicated, but it's still monotone, okay? So we can use theory of monotone dynamic system to show global stability, right? And understand dynamics, okay? But all understanding are based on asymptotic analysis, right? So when we talk to policymakers, all those managers for these lakes, right? They will say how this thing here, these beautiful theorems, relevant to the reality of what they try to control. Okay, yeah, that's a good question, right? So then we go back to this simulation again. Okay, so we just plot some simple solution for this model AQP, right? Okay, yeah, I changed this notation here because it's two different papers, like different notation, but the same model. Okay, so the PhD student, Chris Heglut, had studied this as his core part of his thesis, okay, PhD thesis. So we started looking at a simple solution and to see what matters, okay, what matters for the managers. So would the asymptotic dynamical analysis be helpful? Right, that's the question. Maybe not, right? Maybe not. So we can see the few stages. We just write 1, 2, 3, 4. Okay, first stage, we can see abrupt uptake of phosphorus. So you see cell quarters just shoot up to the ceiling kind of, right? So it's in a very fast time scale. And the light limited bloom state, that's the number 2, right? So it's limited by light, that's the blue color, okay? And red color means limited by nutrient, here's phosphorus. Then we have the crash of this bloom, right? Transition to low algal density, and it's in the process kind of nutrient limiting, right? Nutrient limited, okay, in the transition. So it has collapse of the bloom. Then stage 4, just go back to the low algal density stage, right? So if we look at the algal cells, the density seems like nothing happened, right? Nothing happened. So if we perform asymptotic analysis, nothing happened, right? But the reality is not actually. We have seen bloom, right? Which is a headache thing, right, for the government. So we try to control that bloom, and we need to understand or forecast that, right, ahead. How long, if it occurs, how long it will take, right, and how severe it will be, right? And it seems like they are on different time scales, right? So maybe we can use fast slow dynamics, right? We call this multi-scale analysis to understand transient dynamics and hopefully give us direct insight on these algal blooms and also for the management purposes. Okay, so again, there are a lot of details here. Here, first we replace the equation P by the total phosphorus in the system. That's A times Q plus P, right? So we will have AQS, 3 equations. Then AQS will be non-dimensionalized to get these 3 equations, U, V, W, okay? And using realistic parameter values, we can see the different scales for parameters after non-dimensionalization, right? So that's indicated, okay, delta and beta, right? Kind of small parameters, right? So we're going to handle this equation for fast slow dynamics. So we are going to have the approximation system. Okay. So fast dynamics. Okay. I don't get into details, but just to have 2 scales. Okay. One is fast, one is slow. Okay. And fast dynamics will give us insights on these 2 possible trajectories for the cell quarter. So fast dynamics actually relevant to cell quarter only, right? As we have seen in the simple simulation. So you can see that if cell quarter starts with a bit low ratio, then goes to a level less than 1. Okay, if a bit higher, okay, then finally it gets into the number 1. Okay, number 1. 1 just corresponding to the maximum cell quarter. Okay, maximum cell quarter. Here it's because it's scaled. Okay, scaled, non-dimensionalized. So that's why it gets to 1. Okay. Or we can get slow dynamics. Okay, slow dynamics, and we can get critical manifolds. That's the crucial part, okay, to show the when we have in what kind of situation we have bloom, when we don't have bloom, okay. So we can plot these two critical manifolds. One is called M00, that's the purple plane here, with sufficient nutrient, okay, phosphorus here, but light limiting. And we have M01, the other critical manifold, which is the pink surface here, okay. So that has sufficient light, but nutrient limiting, right? And the double arrow means fast dynamics, and single arrow means slow dynamics. Here we have seen some poor solutions as two possibilities. Okay, so we can see the black curves. They will stay on the purple plane for a while, then crash. Okay, then crash. And the blue dashed curves, those two solutions, you see that they never stay on the purple plane. They just go up and go down immediately. Right? So then we don't see algal bloom. Okay, so that tells when we have the bloom or not, right? So it's kind of we want to use some mathematical tools, right, called fast slow dynamics, dynamical analysis, to show the different scenarios. Okay, when we have the bloom or when we don't have bloom, right? Of course, next question is if we have bloom on the blah blah conditions, what's the longevity? How long it will stay, right? So that's also an interesting question for the policymakers. Right? So we can estimate the longevity, just how long the bloom will stay using different approaches, right? Okay, so if you have interest, you can read the paper in the appendix for details about the approximations, right? They are pretty close, okay, different approximations. So especially I think the explicit approximation is far away. I think that's farthest away from the reality, but it's easy to use, okay, for policymakers, because we just have a formula based on those environmental conditions. You can just plug in values to estimate the longevity, how long the bloom will be. And we also proved that this reduced system, or you can say the approximation system, right, on the fast slow dynamics is a small perturbation of the real system, okay, for system dynamics near the critical manifolds, okay. So those M00 and M01, they are almost consistent, okay, near the critical manifolds to guarantee analysis works, okay, analysis works. And this can be guaranteed by the Fenichel first and second theorems. Okay, that's also in the appendix of the paper. So I think we don't have much tools, right? We don't have many tools to use for transient dynamic analysis. So this is one, okay. So we maybe have some very limited tools, okay, to handle transient dynamics. Yeah. So I think any contribution in future on transient dynamics will be really interesting. Okay. Of course, that's very challenging compared to asymptotic analysis, right, on the limit. Now, next question is if we just we thought, oh, we are very proud to show that we can tell direct insights, right, from model for the blooms and management. Then next question is if you talk to those limnologists, they may ask you why you just assume those kind of light, nutrient are the key. Yeah, possibly, okay, those are the key, right? Very likely we can say that. But they can argue there are so many core values in the environment. For example, temperature, pH value, or even the wind, right? So you can have all kind of conditions and also other conditions like in the landscapes, right? Landscapes near those lakes, right? Maybe those automata and human activities. So there are so many factors we ignore, right? Even though we can say very likely, okay, neutron light are the key culprits for the algal bloom, right? Then next work we did after that, also led by Chris and postdoc Jingjing, and with a group of limnologists and also machine learning experts, we explored this tons of data, okay, from these 78 lakes in Alberta, okay, in Alberta province, in the past 30 years. Okay, so it's kind of long-term data for many lakes, and not just the lake data. They also have the landscape data. Okay, and also so you can see the watershed, right? Watershed like values, right? Values, percentage of pasture land, cropland. So all those watershed data was available from those data sets, and also lake features like lake depths, right? And also, of course, environmental conditions. As I said, like temperature, like precipitation, like waterfall, and wind speed, right? Of course, you can imagine some of this not important, right? Light, solar radiation is light, right? Yeah. Sticky depth, that's relevant to the kind of property of the lake and also the light, right? So how deep you can see from the water, right? The kind of turbidity. Yeah, so there are a lot of factors. We don't list all of them here. Okay, here just example list some of them. So from the so-called incomplete timely data. So we have tons of data and timely data. Incomplete because most days in winter they have no data. Okay, have no data. So obviously we have a lot of missing data. Missing data. Yeah. So I think our algorithm, or we can say we train a Bayesian network. Okay, based on those incomplete timely data to show the probability of blooms. So kind of forecasting. Okay, based on different covariates. Right. So either low or high. Right. So it's a blue one just less likely, right? Very low probability to cause bloom, and the orange ones means very high probability to cause bloom. Of course, if for one covariate, if the low or high give complete opposite result, that can be an important one, right? Can be important one. So from Bayesian network, we can see this graph, which kind of validation for our prior assumption for stoichiometric models. Okay? So we can see the blue ones highlighted are the key covariates to cause the bloom. Okay? So we can see the phosphorus here, nitrogen here, percentage of petroleum is kind of nutrient like input, right? Nutrient input. So it's kind of nutrient part. Current cell count, kind of initial condition, right? And the mass, kind of the timing is relevant to temperature, like light intensity, those things, right? Can also be important. So I think it's kind of validate for the prior assumption, right, for stoichiometric modeling, right? And of course, the machine learning model has some power of forecasting, okay, compared to mechanical models, right? And the disadvantage of the disadvantage of machine learning modeling approach kind of it's hard to understand the mechanisms. Right? Normally, it's not easy. But even this one here just shows some correlation, not causality. Okay, so I think mechanistic model we can use them to test hypothesis, and maybe we can understand some causality. Right? And eventually, I think we need the integration of these different approaches together, as I mentioned in the beginning. And here is the application to the recent project. It's pretty ambitious and it's a pretty big grant supported by federal government in Canada. So we try to understand the methane biogenesis, you can say methane emission, right? So that's I think methane emission is more severe than the carbon dioxide one because that for the global warming effect, methane is more crucial. Okay, that has higher impact than CO2. Okay, than CO2. And here's the data to compare different provinces in Canada for greenhouse gas emission. Okay, clearly our province Alberta is releasing most. Okay, most greenhouse gas. If we compare methane biogenesis, even more severe. Okay, even more severe in Alberta because we have the massive oil sands industry. And this is a starting paper we had. We have experiments and data fitting. I think this is a good example to show the true data fitting. Okay, true is question mark, of course, the quotation mark still, but we can say very likely it's true. Okay? And we can see this trend for these methane emissions in Canada. Seems like the emissions are under control. Okay, under control. They just decays, right? Of course, many people say, "Oh, that's questionable." Right? Questionable. So I think in the very end of this talk, I will just say a paper. We just try to check, or one goal of that paper is to check whether this is true. Okay, it's indeed decaying steadily. Okay. Study area. That's the in the northern portion of Alberta province, where we have tons of oil sands industry spots, right? So we have a lot of oil sand tailings pond and peat lakes there. So they are releasing tons of greenhouse gases. Yeah. So here we focus on methane release. And if we look at the mechanisms for the model, we know something, okay? Even though, as I said, we don't know everything, but we know some key mechanisms for the greenhouse gas emissions, right? So they come from degradations of hydrocarbons a few steps through the microbes, right? Microbes. So we are working on 4 model, but here I just show you the one-step model, okay, for convenience, and also one species of bacteria for convenience. My lab is checking all the DNA sequences of all those bacterial strains, so we can understand their functioning, and we are modeling the more groups of bacteria with different functioning, the biodegradation. Here in starting model, before we didn't have those DNA sequence data, so we had this single species because we have no knowledge, okay, about functioning of different groups. So we just assume they just do the work together, decompose the hydrocarbons, CI. CI means you have a group of hydrocarbons, right? Indicated by this CCHH. Okay, so little c, little h, just two integers, right? Elemental, like the molecular structure, okay, of the hydrocarbon. And we can calculate, this is kind of stoichiometric model. Here we use the minimum function to indicate this Lebesgue law of minimum, that idea, okay, which is non-smooth, okay, in the dynamic system perspective. And GI is the gas release, right? So of course here we use the conservation law as well, right? Conservation law as well. CI for endophyte legs it's 0, okay, but for tailings ponds, CI depends on the dumping rate of the new hydrocarbons from industry, okay. Endophyte legs means they don't dumping any more carbon, right? So that's 0. Okay. And the system should be have conservation law for carbon and for nitrogen here. Okay, so N is nitrogen. Okay, nitrogen. So we have used conservation law here as well, and we can calculate this gamma I as stoichiometric conversion factor from this chemical reaction. Okay, so given the hydrocarbon CCHH, we can compute this gamma I for CH4. We can also compute the stoichiometric conversion factor for the CO2, right? CO2 as well. If we want to calculate CO2, we can do it. Okay, we can do it. But here we focus on the more severe gas. Okay, for global warming, that's CH4. And GI come from the model, and eta I is the kind of conversion efficiency. Okay. Yeah, so here we can kind of mechanically compute the methane release. Okay, methane release as the simplest model I think we proposed from stoichiometric approach. And we take these samples, okay, from the field and take them back into a lab, right, from the mature fine tailings, and take them back into a lab to test those key parameters for degradations. Okay, so there are tons of different hydrocarbons, right? So we need to check them one by one for the parameters to avoid overfitting. Okay. So you can see this, I just gave an example of these degradation curves. We want to check how those bacteria decompose those hydrocarbons, right? So we can get those degradation parameters. So the model has no free parameters, right? And the fitting will be challenging because you need to fit the gas release, right? Gas release. So it's easy to be complete off, right? So if you have interest, you can read our this very first paper, okay, and which has very long supplemental material for the data fitting part, okay, for the details, how we use experiment to check parameters and how we compare with the gas release, okay, the methane release. And we compare with the classical models people use, very simple, so-called zero and first order kinetic models, okay, for the maybe methane release measurement and also prediction. Okay. So we can compare these models to see that sometimes they are all the same, right? They work well for all these models work well for the data. Sometimes some models are off, right? So I think stoichiometric model works fine, right, for most cases. And sometimes the zero order, first order can be complete off. The reason is here, you cannot simply use the least square fitting to get the parameters, so it's easy to fit. That's kind of overfitting, right? But if you have no freedoms, the models can be complete off, right? Complete off. If it's not mechanistic, it's good enough, right? If it's not good enough, then it can be complete off. So it seems like the first order one can catch in most cases, right? But not always, okay? So these ones we can compare statistically to show which one is the best or much better, right? And we also test this against the Synclude MFT case. And even the first order can be complete off for the methane release prediction, okay? So here we cannot call this prediction, it's kind of validation of the model, right? And when the model is validated, we can make the predictions, right? So this approach here, of course, one criticism is, yes, we do very well for the experiments for the model validation, right? But for the field, is that the case? Yeah, that's a very hard question. So I think logically, it's a very challenging scientific question for any modeling. Okay, any modeling. If you can do controlled experiment to show the model is correct, how can you show the model works for the field? Because in the field, there are a lot of uncontrolled covariates. Again, it goes back to the same question as the previous example, right? Stoichiometric model for the algae, right? Similar idea. So maybe you can challenge this still, right? So even though you can fit the experimental data, right? Then that means the model works for the field. Yeah. So that's a very, very challenging question, right? Yeah, then we had this ambitious project granted, right? So actually we started with CRD project. Then we had the Alliance Mission project, which is even larger, right? So I think first one like less than $1 million, but second one is like 1.4 million. So we can expand these experiments dramatically. In many cases, we recruited like a lot of experimental people, right, and also modelers together, try to answer that question, okay, in the connection from the lab to the field, which is challenging. Even you have tons of experiments, still very hard because in the field you cannot manipulate those environmental conditions, right? You can only do that in the lab. How we make the connection? We can collect all the data from field, yes, but you cannot manipulate or control anything in the field. So for causality, that's a very challenging situation, right? Okay, so in the CRD project, we had tested different temperatures, okay, in the lab. So it's kind of gradually we hope, okay, we can understand more and more and more covariates for causality in the lab. But still put them all together in the field, what's the situation? Still challenging, right? So this experiment try to mimic the field case, okay, field case. Along the depths, there are different temperatures, okay, different temperatures. So in the lab, we try to mimic those temperatures to understand causality under control because causality can only be understood under controlled situation. If you compare controlled case with varied case for that factor, right? If you have all varying factors, then you don't know which one is the causing covariate, right? So you don't know that, right? So that's the challenging part. And here is one effort, okay, in very recent last year with a group of postdocs, and I think most of these are postdocs, yeah, and a couple of graduate students in 2005 last year. Okay, so we try to make the connection from field data modeling and so should be the lab data, right? Modeling and field data. Or can be the opposite way, right? Because we have the field observation, we try to understand, then we design experiment and model, right? Try to check the causality. Then how we couple them together, still very challenging. So we try to use some deep learning, try to connect the causality in the lab to the field massive data together. And so that's the effort we made. Of course, still a lot of criticisms. I think it takes time. Okay, still takes time. But I think we need to start with something, okay, to make this connection, even though still have some issues to be resolved. But I think someone need to start, okay, to make these connections, right, through modeling and this big data analysis, right, and the machine learning, right. So I think machine learning AI give us some hope, okay, some hope to make the connection. Otherwise, whatever we do in the causality in the lab, still people can question whether that's the case in the field, right? Still kind of guess. Okay, I think I need to skip this. Okay, it's about the time. So recently, this is another effort. We use satellite data. We know we have tons of satellite data, right, which can give large scale methane release, or you can say measurement in the atmosphere. The issue is the accuracy is low, right? So it's kind of low resolution, low accuracy for this kind of data, but it's massive. Okay, it's massive. It's better than before the data came from those sensors, right? So we can measure those. We can use put sensors in the mining region, for example, to check those release. And in the lab, of course, it's easy. We have GC machine, right? We can measure everything we want, right? But not large scale, right? So if we want to understand huge scale methane release in Canada, in North America, right? So we need satellite data, right? Images, and we use the AI again to extract the data, right? So we need to extract the data extract data from those, you can say, vague images, right? So hope we can extract them from AI, like image analysis. Right? Yeah, so that's what we did in the past year, and yeah, a lot of members working on this ambitious project. And again, this graph here also still the coupling of mechanistic modeling and machine learning, right? And we have a lot of efforts on this, but still in a naive stage. But I think it's promising. Okay, eventually we can make this eventually convincing. Okay, eventually convincing how to connect the controlled lab experiments through modeling and machine learning to the field scenario. Fully understand that, right? I think it takes maybe a couple of decades, I think. Okay, some take-home messages. So I showed the stoichiometric modeling in very quick mode. I think it's not easy for you to fully understand this kind of modeling in short time. I think needing maybe several lectures on this kind of modeling goes through slowly term by term. And I showed that transient dynamics can be more important compared to asymptotic dynamics. The reason we worked mainly on asymptotic dynamics because of we only have math tools on that, right? Very limited tools on transient dynamics. But in many situations, transient dynamics are more important, okay, for scientists. And the true model validation to avoid overfittings. I showed one example. We can have controlled experiments track parameter by parameter, one by one, right? And to avoid the overfitting, but that's normally hard to do, right? Because modelers, I can say probably almost all modelers have no control on experiments, right? So that's hard thing, I think, to do for most modelers. And I showed like a non-smooth system. That's from that Lebesgue minimum, like a monotone dynamic system in that one-page proofs, right? Theorems, and fast low dynamics, perturbation method, or maybe machine learning. Very briefly, all of this. Okay, I never got into details of any of this. Okay, but the point is, okay, maybe in future we need hybrid approaches. Okay, of all these things. Okay, which is in progress. And we have done some efforts, but still need to be improved and more convincing in a lot of situations, I think. Yeah. And greenhouse gas emissions here mainly I talk about methane because that's of course main concern, okay, on the global warming then compared to CO2. Okay. We can also handle CO2 as well, but less important for the estimation, control, mitigation, and also future planning of those industry, right? Okay, so I finish here. Many thanks to all collaborators of this project, of not this project, many projects in this talk, and also current lab members supporting grants. Okay, so I think I finish here. If you have any questions, you can ask me.